Dr. Supriya from Harvard University. So we are about to start our session. Dr. Rudravadi Shahu from Chemistry Department meets Madanpalli will chair this session. So I am inviting him to chair the session.
Good morning to all of you. I would like to introduce Dr. Supriya, one of the eminent speakers in our conference. Dr. Supriya received his PhD in organic chemistry from the Indian Institute of Science in 2014, where he focused on carbohydrate chemistry. Dr. Dey pursued extensive postdoctoral research at prestigious institutions in worldwide. He worked as a postdoctoral researcher at Academia Sinica, Taiwan, the Scripps Research Institute, California. He then joined at Harvard University in the Department of Chemistry and Chemical Biology. Dr. Dey has authored numerous high-impact publications in journals such as JACS, Nature Communication, Chemical Science, Organic Letters, and Tetrahedron Letters. His research has also been recognized through invited talks and keynotes presentation at international conferences, including the European Glycoscience Community, the Canadian Glycomics Network, and the Mid-South Glycoscience Meeting. In addition to his research, he has served as a reviewer for reputed scientific journals and is an associate editorial board member of Current Organic Chemistry. With a strong background in organic synthesis, medicinal chemistry, and glycoscience, Dr. Dey continues to contribute to advancement in drug discovery and therapeutic developments through his work in academia as well as in the pharmaceutical industry. Now, I would like to request Dr. Dey to continue the session. Over to you, sir.
Thank you, Dr. Roy. Good morning, good afternoon, and good evening. Thanks for the kind introduction. And my apology for all this computer glitch and other things happened during last 10-15 minutes. So today I'm going to present my postdoctoral work with Dr. C.H. Wang, initially at Academia Sinica, Taiwan, and later at Scripps. And my talk is entitled as Programmable one-point synthesis of heparin sulfate and heparin-derived commercial anticoagulants. Before I start, my sincere thanks to Dr. Esmi Rai. We are connected like 15 years, but we met a couple of times. And our connection is based on our background on carbohydrate chemistry. So when she was in PhD and I was doing PhD IIC, she was in IIT Kanpur. We had connection at the time, and then we met in ICS 2014, almost 11 years back. So thank you for this invitation. And if you go to the next slide. So as I mentioned, this work carried out with Professor Chiu Wang. So I joined Wang Lab in Academia Sinica first in Taiwan and later moved to Scripps. So I'll be talking how we developed a programmable one-part synthesis to synthesize commercial heparin sulfate and heparin-derived anticoagulants as well as some glycans. Next slide. Yeah, so I think yesterday you have a essence about the glycoscience and carbohydrate molecule by Professor Bhankar. So you know now many of those how the sugar structure look like, how it is galactose, glucose, mannose, ribose, arabinose, and other things. So in Wang Lab, when I joined Wang Lab, I was majorly interested in the cell surface glycan, specifically glycosamine glycan. So you know the surface of the glycan, surface of the cell is like M&M and chocolate. So you can see that the inside is a lipid layer, bilayer, and outside is coated with the sugar. So we have this different kind of glycan in the surface of the cell, and they are responsible for many functions, and major importantly is sialic acid. If you see sialic acid, viral infection, flu, rotavirus, this all mediated by the sialic acid. And this glycan is divided into two major categories. One is glycan, which is non-charged, as you can see here. It can be called either N-glycan or O-glycan, depending upon the linkage with the glycopeptide. And then you also have glycosamine glycan, which is charged. And it's very important, the linkage as well as the pattern of the glycan is very crucial to underlying the disease or to identify the disease. As for example, in cancer cell, you can always find there is a high expression of the sialidase and sialotransferase, and you get the highly sialic acid containing glycan. So if you may remember, we have come through this difficult phase of COVID-19, and then from CCRC in Georgia, somebody has found out that you know viral protein has this kind of glycosylation site, and then majorly they have found high mannose glycan and then O glycan and complex type glycan. And the linkage is very important. Like, so even if small changes in the structure of sugar in the periphery can differentiate a lot of things. Like in case of sialic acid, when it's linked to alpha 2,3, is avian and swine flu. For human case, is alpha 2,6. So generally, that's what is very difficult. COVID is one case, but to transmitted avian flu to human body. Next slide, please. So this is how in glycobiology, how this define the color coding. You can see this in one of this N glycan structure. This is in bisecting with the fucose at core, and then you can see the different color coding. So galactose is colored as I think yellow, mannose is green, this galacnac, and then fucose, sialic acid. And in glycosamine glycan, they have a different color coding. I have not put here, but you can see in subsequent slide. But there's four major kind of glycosamine glycan: is heparin, creatine sulfate, dermatine sulfate, and hyaluronic acid. And this you can find in many of eye drop, some skin product, everything. You can see this. If you see the content of any of those, you can find hyaluronic acid. And heparin has been used for anticoagulant for a long time. KS is very important for different development in eye, and dermatine sulfate has a different role in different tissue. So we go to the next slide. So so far there's two approved drugs by FDA on anticoagulant, heparin anticoagulant. One is fondaparinux, which was approved by FDA in 2002, and then there is second version of that, which is iodoprenix, which is methylated version of this pentasaccharide motif. So this anticoagulant basically do clean the clot from deep vein thrombosis and pulmonary embolism. So if you have a clot in your vein, this can drug these both fondaparinux, iodoprenix, as well as low molecular weight heparin sulfate also can clean the clot. And if you read many of these news articles, specifically New York Times and Washington Times during the COVID, so they found this kind of difficulties in pulmonary embolism in the COVID patient, and they recommend to use the fondaparinux to clear the clot. So heparin and heparin sulfate has been used as an anticoagulant, but this is not only the potential properties they have, but now there have been a lot of work has been going on in different academic and industrial lab settings, that how this kind of complex glycosamine glycan molecule can be also involved in deaggregating the toxic tau, which is a crucial complex for the A beta protein and Alzheimer's disease. And there's a nice article, I think, from I forget it's GJ Booms or somebody else lab, that how the COVID SARS virus use HSPG to do endocytosis in the mucous cell. If you go to the next slide, please. So now the trouble is, like, we understand that heparin is crucial. This has a lot of properties as anticoagulant and can be readily used for different therapeutic application. But the challenge is like how to get the heparin, right? So that's a big challenge. So high demand of pharmaceutical heparin, and then only mammalian heparin is the major source, and then this production, major production in China, and then use pig gut to isolate heparin. And the issue of quality control for commercial heparin, difficult to separate contamination and heparin. And there's one example back in 2008, 100 patients died in USA due to the contaminated heparin sulfate. And I think Bob Langer and others, other few folks in MIT, they have done a thorough work, NMR investigation, to find out what is the exact contamination. And you can see this contamination here listed. This is a nice paper published in New England Journal of Medicine 2008. So you can see even it's over sulfation and chondroitin sulfate mixed with the heparin sulfate can give a deadly combination. So now question is, what is the solution? So solution is that you can use marine organism as a source of heparin, and then chemical synthesis. But chemical synthesis is lengthy, and because you can see the structure of heparin in the last slide, that we have multiple sugar units. We have 2-amino sugar. We have other iodose sugar. So the reactivity is very different. And then you have to have alpha selective glycosylation, specific selective glycosylation. You have to install regioselective sulfation, N-sulfation, O-sulfation. That increase the synthetic step with multiple purification, low heat, efficiency, and that high price. So as per 2020 data, there's 12 billion market in heparin in industry in US. So possible solution maybe we can do automated type of synthesis. We can do enzymatic synthesis. We can now shrimp head. A particular class of shrimp head has been used to isolate heparin in comparison to pig gut. Can you go to the next slide, please? Yeah, so when I joined Wang Lab, we are thinking to develop a programmable one-part synthesis, which has been developed by Professor Wang back in 1999. But we want to optimize a new software to try to ease the synthesis of this complex lichen. And if I go back to see how this development of programmable one-part synthesis, I just try to provide here a brief history. It based on the reactivity, is the kinetics of different sugar and how they couple to each other. Hans Paulson back in 1974 in Germany, he has found that protecting group affects the rate of hydrolysis of glycosyl chloride. And then if you're a sugar chemist, you of course know that Fraser-Reed, arm and research concept, which has been developed back in late 1980s by Robert Muto and Fraser-Reed together. And then mid-90s, Stephen Lay tried to optimize the synthesis of complex oligosaccharide via reactivity quantification of donor. And one of these paper, I think in Syncom or I don't recall, in 1995, he stated that in order to achieve the synthesis of complex oligosaccharide, the reactivity of donor must be better quantified. And then late 90s, Dr. Wang and co-workers developed a method called relative reactivity value, or RRV. It's based on 1H, proton NMR, HPLC, and computer program at the time. So the old software is known as Optima software, and we have developed a new software called AutoCho back in 2018. So how the software work is, I provide is a simplest workflow. So if you have a glycan structure, you can input the glycan structure, and the software give you output. Is basically do the retrosynthesis, that how you couple those molecules in sequence, and then you synthesize those molecules by enzymatic synthesis or chemical synthesis, and then you couple them as guided by the software, and you get the protected oligosaccharide. You deprotect it and get the desired oligosaccharide. Now, difference between the AutoCho software and Optima software is that in Optima software we have they have before measured the relative reactivity RRV for 150 glycosyl donor, but they don't have like a big library. So in AutoCho software, we try to attempt to make it more computed. So you don't need to measure the reactivity each and every thioglycoside donor you want to use. Rather, you can predict the reactivity. So to validate this, we collaborated with the Institute of Computer Science in Academia Sinica, and they come up with a method. Then you can predict the reactivity of thioglycoside donor using a SBM regression model and a lot of, I think, computer term. I'm not going to detail on that. But the key point is that if you see the predicted and observed reactivity is very closely matched. So now the advantage of the AutoCho software is that we have a large library of 50,000 building block reactivity, and we can measure it. So by theoretically, and only need to measure every single individual by chemical synthesis and then measurement of RLP. If you're more interested, please look this particular GOV article. We have its video. You can find out that what exactly the AutoCho software do to ease the synthesis of complex oligosaccharide. Next slide, please. So this AutoCho software use graphical user interface, Java runtime environment, and now, as I mentioned, we have more building block with the validated reactivity. We can accurately predict the reactivity with the virtual building block. That means we don't need to synthesize those building block, but building block does exist. And we can do fragment coupling, and we use chemical shift, and then they use the SVM regression model for RRP predictor. You can input the structure by mouse drawing, sugar type, anomic configuration, define the fragment to be used. So here is one example. You may know that TACA, tumor-associated antigen, cancer antigen, GloboH, the structure of GloboH, and if you put the structure in AutoCHO software provide you the fragment that this glycoside donor and acceptor can be combined in particular fashion to get the desired GloboH, which was later went to the phase 2 clinical trial for the cancer vaccine from Wang Lab. Can you go to the next slide, please? So now question is how you measure the reactivity, right? We understand the reactivity. So how we measure the reactivity? So the reactivity measurement has been performed by Wang group back in 1999 in the first paper. So say you have two thioglycoside donor. One is you don't have the reactivity, and other you know the reactivity. So you make the reactivity 1, which is pentacetate estolyl glucose. And then you treat it with the methanol and then use NIS triplic acid DGCM in room temperature to make sure that the activity of the glycoside donor is optimal. So that means all your acceptor is reacting with the donor. And then you quench the reaction in half an hour, filter it, work up, and inject this in the HPLC and find out the absorbance of the reference thioglycoside donor as well as the standard thioglycoside donor. And you do this, and then you dissolve those two thioglycoside donors separate in DCM and inject the HPLC to get the absorbance. So if you have all the absorbance, if you put in this equation, you can get the D reference, which is the reactivity of this particular thioglycoside donor. So you can measure the reactivity. So once we developed the AutoCHO software, I was more interested in the heparin sulfate, and then I put this pentasaccharide motif, and then AutoCHO give the output that this can be synthesized either compound 1, 2A, and 2B, or compound 1, 2A, and 3, and compound 1, 2B, and 3. Now, if you see, interestingly, because this cycle only works when you have reactivity in the decreasing order. So you have to add those in sequence. So R1, 2A, and then 3, 1, 2B, and 3. So you cannot add like 2B, 1, and 3. It will not work. So it is in parts. It is based on the reactivity. So sequential addition of this thioglycoside donor, and this acceptor is, you can see this is methoxy, right? So it is no donor. So that is no reactivity. That's what the reactivity is 0. So basically 1, 2A, and 3, and 1, 2B, and 3. And then you do 1-port glycosylation, and then you do deprotection and chemical sulfation to get the desired pentasaccharide. Next slide, please. So I'm not going to show the synthesis of monosaccharide and other thing, but I will show some steps which we face little difficulties. So this compound 26 and 27, where we have R group as TBDPS and PMB, as you have seen before that we have to make 2A and 2B as part of the guideline from the AutoCHO, and then we react with 28, which is 2-azidoglycosyl donor acceptor, and then you react and they get disaccharide, and then remove the livolyl group and to get this OH compound 4-hydroxy, and then measure the reactivity, which has OH and then a PMB group at the 6%, and reactivity was 38.2. And we also have the TBDPS group at 6%. Now we have to oxidize, as per the structure of the heparin, we have to oxidize this disaccharide to make this CO2Me at 6%. So we took compound number 31, remove the TBDPS group, and then try to oxidize with BIB tempo or tempo KBr, but we did not get any oxidized product. But however, when we remove the 4% livolyl group and then try to oxidize, we got this compound. So I don't know exactly what kind of role that 4% livolyl group plays, but if you get dihydroxyl compound, you just put BIB tempo, normal condition, and then get acid, and then in situ esterification, and then you purify, you get compound 2A, and we measure the reactivity. Now, if you see the reactivity of 2B and 2A, right? So 2A is less. So even the reactivity is in the first, this anomeric center of your reducing in sugar, but the non-reducing in sugar has some impact. So this has changed the reactivity. That I think conversion of this ester provided some reduction of the reactivity. But nevertheless, we got 2A and 2B. Next slide, please. And we face these difficulties when we try to couple iodos with this 2-azido 2-azidoglucosyl donor. So we have TBDPS and PMB group, a mock group, and try to glycosylate, we get alpha beta mixture. We are not very sure because even though you have OBZ at C2, you should get neighboring group participation to get the desired isomer, but not. But when you oxidize the C6 position and to esterify and start with this, we eventually get only alpha configuration. And once we got these three compound, I will show in the next slide how we do one-port glycosylation. But there's another problem we faced when we have this pentasaccharide motif and we want to do saponification and O-sulfation. And what happened is, if you have a TBDPS group, you want to try to saponification and do the O-sulfation using sulfur trioxide pyridine, these will incorporate it inside this particular structure. So then we have to change our protocol, remove the TBDPS group using HA pyridine, and then do O-benzylation. Now, O-benzylation was also very difficult because you have multiple base level group. So we cannot use any sodium hydride. We tried silver oxide in BR in toluene, the yield was low, and then we use this particular solvent combination in seal tube at 80 degrees centigrade. We got a desired compound. But before going to the deprotection, I'll go the synthesis of one-part synthesis of the heparin sulfate. You go to the next slide, please. So as I mentioned before, that it has to be sequential. So compound 1, 2A, and then you can take any of this donor except 3A to 3C, 3A, 3B, 3C, 3D. So first, and you can see this pentasaccharide motif. Now, we used both 2A and 2B as guided from AutoCho software. Now, interesting part is that you get all alpha glycosylation because that is the requirement of the pentasaccharide motif. And this alpha glycosylation is governed by presence of TBDPS and acetyl group at the C6 position, which just block the beta phase so that incoming nucleophile should attack on the alpha phase. Now, question is that the computer AutoCho program give two method, right? Two different route. Which route is now? For 2A compounds, you have already ester in place, so we no need to deprotect and make ester. But for 2B, we need to deprotect the PMB group, which we did by DDQ and followed by the tempo oxidation and then esterification. So this multiple step reduce the synthetic yield, but as far as prediction from AutoCho, both works very well. So it doesn't matter. But for us, compound 1, 2A, and either 3A to D was more preferable because we no need to do additional 2 steps. And at the time, we predicted, as I mentioned, that TBDPS and acetyl group can block the beta phase, so incoming nucleophile from alpha phase. We did not publish as a methodology, but we're pleased that after 2 years of our publication, group from China, they have shown that the same concept, that you have a OBZ group or this halal group, you can basically block the beta phase and incoming glycosylation for 2-azido sugar come from only alpha phase, you get alpha product. If you go to the next slide, please. So I have shown the problem difficulties with the presence of TBDPS to do saponification reaction and how we optimize the benzylation reaction. So now we got this pentasaccharide motif. So remove the TBDPS group here and then got hydroxy group using HA pyridine and followed by established protocol with the benzyl bromide silver oxide. We got this pentasaccharide motif. And then we use saponification reaction and followed by so when you do saponification reaction and followed by O-sulfation, you get O-sulfated and then ester get hydrolyzed, and then followed by reduction of N3 to amine and then selective N-sulfation provide you the pentasaccharide motif. Now, interesting fact, this is the first example when we functionalize heparin 6 sulfate with the regio-defined sulfation pattern. So you can see that compound 1A has 3 hydroxyl group primary, but we can modify the C and E fragment, keeping the A fragment as hydroxy. And as the beginning, I mentioning the key, right? How you use that glycobiology key. So this is how they use for glycosamine glycan. So in glycan case, it's a single color, but glycosamine glycan you get like this kind of settings. If you go to the next slide, please. And similarly, if you want to make one sulfate, we do the same protocol. We remove again some protection chemistry, remove the labelyl group, followed by benzylation and similar step. We get only one sulfated pentasaccharide motif. So why now? Question is why we synthesize this molecule? Because we are interested at the time. I think some studies has been ongoing now that how this which particular sulfate interact with the A beta protein and what kind of interaction they have. So we try to understand that you need 1 sulfate, 2 sulfate in the 6 position, because it has been known by S.C. Hung that 6' sulfate has some role to interact with the beta protein, and they have the NMR data on that. So next slide, please. So now question is how we detect this kind of complex, complex oligosaccharide. So it's very difficult, because you see many peak. You have to do all kind of 2D experiment. But most handy was coupled HXQC experiment, because when we send the paper, it is like mandatory to send this kind of NMR data to know that how many alpha and beta is there, and whatever you are claiming that you have that perfect structure of heparin, how do you confirm? So it's very quick test. Like this particular pentasaccharide, we have 4 alpha and 1 beta. So it is known that if your the coupling constant is more than 165 is alpha, and then less than 165 is beta. And we measure the distance between the contour and then coupling constant. We found that you can see that more than 165, 4, that is alpha, and 165, 1 less, that is beta. So we confirm this one finally, that you have basically has selective glycosylation, and you get only 4 alpha and 1 beta as required for the heparin structure. Next slide, please. So once we have optimized the use of AutoCho software for heparin synthesis, we use the same methodology, the AutoCho software for commercial anticoagulant fondaparinix. In the beginning of my talk, I mentioned that fondaparinix is approved by FDA back in 2001 and 2002, and it is administered by sub-Q. And limited dose is 7.5 milligram, and so you can get a kind of single injection there, and its cost is quite high. And fortunate or unfortunate, the only source is chemical synthesis, so you cannot, there's no other sources that heparin fondaparinix can be synthesized or isolated. So before our paper has published, there is few method reported in the literature, and in majority of these they have the low yield, and then the major issue is the stereoselective glycosylation is there or not. So we thought that can we use this AutoCho software and get some improvement in this field. So in our case, we got alpha selective glycosylation as we optimized in as I showed in last slide by TBTPS and acetyl group as C6 position, improve the yield, reduce the synthetic route. So when we input the structure of fondaparinix, AutoCho again give this outcome that you can synthesize compound 2, 10, and either compound 17 and 18, and then you do this similar kind of one-pot fashion, and you can get this pentasaccharide model. Next slide, please. So I just want to go through some of the synthesis. So we start with this dichloroacetamidate, and then we have this synthesized monosaccharide acceptor 4, and then couple them, and then followed by removal of acetyl group and then benzylation group, you get compound 8, and then DDQ removal of the NAP, we get compound 8, and then removal of the TBTPS group and followed by acetylation provide compound 9. And removal of benzylidine group oxidation and esterification provide compound 10, and we measure the reactivity is 2.14. And for iodose, this acceptor, we start with diacetone glucose, and then we can, by known method, we can synthesize compound 12, and then using the same chemistry, you basically do the glycosylation followed by BIB tempo oxidation, and then esterification provided compound 17 and 18. Only difference is that we provided 2 protecting group, one is benzyl and one is acetyl. And I will describe why. Next slide. Yeah, one more time. Yeah. So first we try, we try compound 1 and 10. So it's same concept again, that more reactive, least reactive, and lowest reactive. So 1, 10, and compound 17, and we get only 26% of yield. Can you click again, please? Yeah, but when we change the acetyl group here, somehow we improve the yield by double. So 26 become almost 50%. We don't know the exact reason, like what acetyl do and what OBZ group doesn't do. And if you click one more time, and then we use heparin to remove the TBDPS group, followed by saponification reaction, O-sulfation reaction, and then palladium charcoal to reduce the amine to azide to amine. We get this pentasaccharide motif, and then selective O-sulfation. We got this fondaparinix. So this is, as I mentioned, this is alpha selective, first selective synthesis of fondaparinix, where you have shortest route, you have improved yield, overall yield, but still lot long way to go. We did this synthesis in I think 100 milligram scale, but we also improve by like we can do in 1 gram scale, so it is scalable. A lot of effort you can find online, manuscript, patent, a lot of still ongoing effort how to use the synthesis of this complex commercial drug. So if you go to the next slide, please. Yeah, so there's another one, right, approved by FDA in 2008, hydroponics, which is introduced by Sanofi. So the difference between that, this is not a natural heparin. So they have removed the NH N sulfate and put it as the methoxide OME. And the Sanofi discontinued hydroponics due to the interclanal bleeding of some patient, and then Sanofi introduced a second generation of this drug, is a biotin conjugate of hydroponics to selective withdrawal of the drug in case of any adverse effect. So before us there is 5 report, and again the major trouble is like no stereoselective glycosylation alpha beta mixture in the key step. So we took the challenge and we used again same one pot strategy, and we got alpha selective glycosylation high yield, and we got 6% overall yield in the shortest route compared to the route mentioned in the literature. If you go to the next slide, please. So if you see the literature procedure, you can find is always trouble with the selectivity, right? You get alpha and beta mixture, and then you need to separate. As for example, 7:1, here you can see like 1:1, here is multiple mixture. So that's big trouble when your key glycosylation step has this kind of issue. So you go to next slide, please. So we little bit twist here. I'll tell the story a little bit later, but we started with this pentacetyl glucose and then depotate acetyl group, and then introduced TBTPS group and methoxy group, and followed by convert this acetyl group to phosphate donor. And for the disaccharide, we use commercial level D-cellobiose, and then this is known compound cellobiose to compound 6 is known, and then from compound 6 we converted this thioglycoside donor 7, followed by protection deprotection chemistry majorly cyclic protecting group, followed by TBTPS introduction, benzyl protection. It's tedious, but there's no other way. And then removal of this cyclic protecting group, oxidation esterification, we got this compound 12. And then removal of TBTPS group using HA-pyridine and followed by selective acetylation, we get compound 13. So there is kinetic control acetylation. So if you use more equivalent or you increase the temperature, you get both 4 and 6 acetylated compound. But we have optimized, so we got compound 13. If we go next slide, please. And for this acceptor, we started with diacetylene glucose, we got compound 15, and then we install acetyl group, but we cannot separate at this stage. And then we remove the acetyl group, and we do separation alpha beta, and then took one of this one of this isomer, and then converted to BZ, followed by glycosylation compound 19, get compound 20. And then removal of the OBZ group, introduce OME group, and then same hydrolysis, oxidation and esterification provided compound 22, and this reactivity is 0 because there's no thioglycoside. You go next slide, please. So we start first this one pot method like compound 4, 13, and 22, but we did not get any pentasaccharide motif. We're not sure why, and we got only trisaccharide, but I don't know, like methoxy group has any role. We did not investigate. We did not have any time at the time to investigate the role of the protecting group. But if you click one more time, yeah. But when you introduce phosphate donor, what happened is you use TMS or triplet first to activate this phosphate donor to get trisaccharide, and then you add NIS along with the compound 22, get this pentasaccharide motif. Again, this is 100% alpha stereoselective because of TBDPS and acetyl group. If you click one more time, and then followed by removal of the silyl group, and then saponification reduction, and then global O sulfation provided the synthesis of hydroponics. So this is the first stereoselective synthesis of hydroponics. There is no alpha beta mixture. This is much more straightforward. The synthesis of disaccharide from cellobiose was very crucial to reduce the number of synthetic steps. Next slide, please. So last few slides, I want to show about how we incorporated this AutoChoose software and this methodology for CLDase resistance oligosaccharide antibody. So in monoclonal antibody, only that Fc gamma region always have a glycan, and that glycan is like not homogeneous glycan and heterogeneous glycan. But that glycan is very important to keep the 2D and 3D structure of this antibody. So you can always digest the glycan, but you cannot remove the last, this one, the glucan. You cannot remove it. So we are interested that increase the half-life time of monoclonal antibody, and it is already known that because of the terminal sialic acid, in case of sialidase that can cleave, and then the efficacy and then half-life time also decrease. So we're interested how to increase the stability, study prolong half-life of the antibody, and also can use this particular methodology. And now they have in Wang Lab they published that you can selectively deliver API to targeted organ. So we took a methodology that can we introduce a 2-fluorosialic acid to increase the stability of the sialic acid galactose galactose bond. If you go to the next slide, please. So to do that, there is a complex synthesis because sialic acid donor is very, very low reactive. Who worked on sialic acid, they know that how difficult it is. And so we started with that NPACF3. This is a kind of donor, and then fluoro donor, and then we synthesize this benzyl compound with that galactose, and we try to glycosylate only starting material. So we thought because the donor is poorly reactive, so we cannot get any desired compound. So we thought an alternative route to introduce fluoro. We thought like, can we do a hydroxy at equatorial position and make the sialic acid at the terminal and galactose, and then convert the equatorial hydroxide to axial fluoro. So start with the sialic acid, then convert with the glycal, DMO epoxidation, and then make the bromide donor, and then glycosylation with the hydroxy compound or get this compound 9 disaccharide. Now conversion of equatorial hydroxyl at 3 position of the sialic acid to axial fluoro is nightmare. So we tried many fluorating agents and nothing was helpful. And we found, if you see the Buchwald from MIT, he has used this particular NFF for many of his coupling reagent, and we don't know what is the particular role, but somehow we thought this may be helpful. This is a wild guess. And in toluene DBU, it is a long time, but later we optimize. If you have sequential addition, we can get conversion of hydroxide, equatorial hydroxide to axial fluoro, and we're lucky to get the crystal structure to confirm that. Next slide, please. So as I mentioned, we have used many fluorating agents. AB Dual Lab, before at Princeton, she has been developing fluorating agent and many other labs, but yeah, nothing helps except that particular agent. Next slide, please. Next slide, please. Yeah, so I'm not going to show all this complex synthesis, but then we synthesize compound 1, 2, and 3, and do again 1 for synthesis to get this hexasaccharide. From hexasaccharide, we can get this particular glycan. Now in the glycan, we have terminal sialic acid derivatized with the fluorosialic acid. So when we take commercial antibody, we digest the glycan with N-S and N-2, and then we get, as I mentioned, that we can digest until the last glucanate 1. And using N-2, we can introduce this this synthetic glycan here. So how do you observe that this in vivo study showed that the stability has been increased, and then without efficacy also improve or keep intact, but stability has been widely increased. And we published this one few years back now, but recently Wang Lab has published that you can attach kind of API to your drug molecule in particular fashion and cleave using enzyme in particular tissue target, and you can deliver API in the particular tissue. So next slide, please. Yeah. So last couple of slides, I want to show that I also work on the vaccine development, synthetic vaccine development, majorly for HIV in Wang Lab. So Wang Lab is known for synthetic carbohydrate vaccine for cancer. It was in phase 2 clinical trial back in 2017. And this is the example. We have the GlobeH TACA, and then they have the carrier protein adjuvant. And this particular construct was in phase 2 clinical trial. Now, challenge for the synthetic glycan is that the crisis of like how you make those glycan, right? It's very difficult to make those glycan efficiently. and then conjugate of the glycan, then find out the carriers, adjuvant, and then also the major trouble is carbohydrate in synthetic glycan is like how to switch IgM to IgG, because you need to get long-term T cell response. So that is the main trouble. A lot of work has been done, still ongoing, that how you can increase the IgG. But for me as a synthetic chemist, we are trying to find out how we can ease the synthesis of this complex glycan in the time-effective manner. If you start the chemical synthesis, you cannot get in. Like it's thousands of glycan, right? So in HIV case, we mapped all the glycan and we found out that all glycan has a trisaccharide motif. One mannose is connected with the two glucnet, beta 2, beta 6, or beta 2, beta 4 linkage. So we thought that can we synthesize only this trisaccharide motif, and then can you use chemoenzymatic synthesis to get all the glycan much efficient and time-saving manner. If you go next slide, please. Yeah, so I synthesized compound 2A and 4 and then try to couple. It did not couple, so compound 4 and 8, we get compound 9, and then use EDA and butanol to remove the phthalimide and then convert it to NHSC, followed by the reduction of this removal of the benzyl group to get the compound 11. Similarly, you can get compound 14. The difference between 14 and 11 is that one is beta 2, beta 4, beta 6 linked with the mannose, one is beta 2, beta 4 linked with the mannose, two glucnet linked, the two different fashion. So if you go to the next slide, please. So then I took 2 or 3 months training to how to isolate enzyme, but you can buy glyphosate transferase. So we took commercially engineering E. coli, and then you can express the E. coli, purify the E. coli. So for my reaction, I need alpha 2,6 salic transferase, 2,3 salic transferase, and GALT. So we got those enzyme in good yield, and then for a reaction we took compound 3, the trisaccharide, and using beta 1 per GALT, you can selectively install characters, and followed by alpha 2,3 salic acid, you can get this pentasaccharide. Now, take-home message from here is that even you have acetate protecting group that is acceptable by galactoside transferase and alpha 2,3 salic transferase. So this is the example, and after that we have synthesized many of this glycan motif, and then later has been used for the interaction with the GP120 for HIV. Next slide, please. With that, I'll close here. I thank Dr. Wang to give me opportunity to work in complex oligosaccharide and chemoenzymatic synthesis of glycan. And I have not shown any work from George's lab. I was not doing much of chemistry, majorly engineering stuff in Whiteside lab. And I have not shown any of my PhD work with Professor Jaraman. And I feel fortunate, very fortunate to get connected with Professor Feza Reed back in 2009 when he came ISC, and we become friends. And he used to taught me carbohydrate chemistry. I was when I joined PhD, I don't know which is glucose, galactose. I was very confused. But Feza, I think he taught me everything. He passed away during COVID time. And thanks again, Dr. Roy and MITS for your invitation. And happy to take any question if you have. Thank you. Thank you for kind attention. Thank you very much. I don't know, I take a little longer or not, but thank you.
Okay, now the session is open for discussion. If I am requesting our audience, if you have any question, please go ahead. I have one question, Dr. Supriya. Yeah, you made that heparin sulfate molecule. It contains a lot of sulfate groups.
Yep.
So how is the solubility of that material, and how did you purify that material?
Purification was a little bit nightmare because in Wang lab we don't have at that time any automated system, so we use Sepharex G25, G25X, G20.
Okay.
And then we use auto sampler to collect this sample and then evaporate the sample, and then mass effect also you get fragmented. So yeah, so we spend a lot of time on purification, final step purification, and then analysis of the compound. Yeah, but this is
and your final mass, that is you took multi-mass or normal HRMS? What kind of mass?
HRMS you get fragmented. So if you see the report, like they report like M-3H plus 4 sodium by 3. So you can find out. it is.
Yeah.
And now I think there is now technique that even in siRNA and this complex heparin and other oligo, they have a software, they can tell you, oh, this is this particular fragment, this is the structure. So now I have not used that, but yeah, you can do that.
I think my advisor, Professor Bhankar, has one question. Sir, please go
Yeah,
ahead.
yeah, it was a
Yeah.
good talk, and I
Thank you.
was very much interested in this talk. I'm not very sure whether I met you in Professor Jayaraman's lab in ISA Bangalore. I visited many times.
Yeah, it's long time back, 2009-10. Yeah.
Yeah. Anyway, the question is that this is related to the benzylation step that you did it when heparin pentasaccharide monosulfation, that
Mhm.
particular. And
Yeah.
the condition that you use is looks very harsh with silver oxide and 70 degrees sealed tube.
Mhm.
So I'm just wondering if you have to do on a little bit larger scale. I'm sure that is a little bit old work, so there may have been some more modifications on that. But what do you think about this step? It looks a little bit because you'll have to use one equivalent of silver oxide or maybe more
Yeah. Yeah. So
to remove the bromide.
yeah, that's the one challenge, definitely, because the silver oxide benzylation doesn't work that well. So when I used toluene, it was like 35-40% yield, and then there's a paper by some Japanese group, and they have thoroughly investigated the benzylation with the different solvent. So we took that particular methodology published in JOC back in 2018. Yeah, but for me, I did like in milligram scale, I think 800 or 900 or 900 milligram, 800 milligram. But I don't feel any difficulties. The only trouble is that you are heating DCM and hexane in seal tube in 80 degrees
Yeah.
centigrade. So yeah, that's the only trouble I got. But interestingly, if you remove one of the solvent, we did not get any product. So I have no clue why this particular combination works very well. That has been published by Japanese co-author.
Yeah, that's fine. Thank you. Okay.
Yes, yes, and your final compounds, when you are taking NMR, which solvent system you are dissolving it, DMSO or what?
This is water also. Yeah.
Water also.
Yeah, and then
Yeah.
if you want, that's what we in the protecting stage we just find out everything, and then final stage only we see the N sulfation, O sulfation happened or not. So I recall in scripts I use a 800 megahertz NMR, and which is only supposed to use by protein chemist. So Laura, the person in charge, she took like was the auto simming. We
Yeah.
started 9:30. It took like long time to sim the auto simming.
Yeah. Yeah.
So it took long time to sim that one, and I have the picture that when I took the NMR. But
And
yes,
and how much amount you got your final material
so
in
when we do
what?
that, yeah, 25 milligram, 25 milligram. But we have done large scale
Oh,
later
that is
on.
good.
Yeah,
Yeah.
we large later on we thought that it could be, especially for contemporaries, that we can be used for commercially. We can patent or something. But after that, I don't know now status. Like he's actively pursuing glycan or he not. So yeah, but yeah, 25 milligram final compound.
Yeah, that is a good yield actually. Yeah.
But
And
purification
so
was good.
if
Normally,
yeah, because in final step, carbohydrate chemists after synthesis, synthesizing the polysaccharides, they are getting means maybe 1 milligram,
yeah.
less than 1 milligram. Then we don't have data for taking NMR. Means this kind of problems
Yeah.
are always associated with it. Anyway, thank you, Dr. Supriya De, for this wonderful talk. Now. If no one has any question, no more question, then I from this organizing committee, I thank Dr. Supriya De for your time and presentation and for this nice talk.